Welcome to today's Explainer. Today, we're diving into a reality where scientists aren't just studying nature anymore. They're actively treating the fundamental building blocks of life as a reprogrammable circuit board. And hey, this isn't just some abstract academic theory. The ability to architect life at the molecular level is literally solving some of humanity's biggest problems right now, from highly targeted disease therapies to drought-resistant crops. So if you've ever wondered how we went from just, passively observing inheritance to actively engineering biology, you are definitely in the right place.
Okay, think of the classic DNA double helix as a piece of hardware, like an electronic circuit board. It's a perfect metaphor for the massive paradigm shift we're experiencing in science. For pretty much all of human history, we were just reading genetics, right? We just watched natural traits get passed down from generation to generation. But now, we're writing the code. We're actively architecting life, treating genes as programmable inputs and outputs that we can tweak, rewrite, or even entirely replace to change how a living system fundamentally operates.
To navigate all of this, our agenda today covers editing the biological blueprint, the traditional transgenesis toolkit, building recombinant DNA, transforming cells and plants, the CRISPR precision upgrade, and we'll wrap it all up with the arc of innovation. Let's kick things off with Section 1, editing the biological blueprint, because before we can manipulate anything, we have to understand the baseline. By definition, genetic engineering is the manipulation of an organism's existing genetic architecture using recombinant DNA, or rDNA technology, to produce entirely novel traits. At its core, we're talking about the artificial transfer of gene fragments from one organism to another. We're treating genes like modular, hot-swappable components. It's kind of like, taking a natural frost resistance gene from a deep-sea fish and just seamlessly dropping it into a tomato plant so that crop can survive freezing winter. What's really fascinating here is our primary output. We take a baseline standard cell with its natural DNA intact. Then we intervene, creating a recombinant DNA molecule by successfully fusing DNA segments from completely different species. The sheer biological mashup of this is just wild when you stop and think about it, and it only works because DNA is a universal language. That shared programming means a specific instruction manual from a bacteria can function perfectly inside a plant or an animal. Moving on to Section 2, the traditional transgenesis toolkit. Because just like any high-end manufacturing process, editing these biological blueprints requires a very specific set of hardware. Think of a genetic engineer's workbench like a mechanic's garage. Our crucial hardware includes 7 specific tools. First, the gene of interest. That's the brand new engine we want to install. Next, we need a vector, which is basically the delivery truck to carry that engine into the cell. We use restriction enzymes as precision molecular scissors to cut the existing DNA, and ligase acts as the biological glue to weld it all back together. We also need a host to act as a factory, a marker gene to verify which cells actually took the new code, and tissue culture, the controlled environment where we grow these modified cells into full organisms. All right, Section 3, building recombinant DNA. Let's look at the actual molecular mechanics of how these tools assemble a new sequence. We can actually break this complex assembly down into 3 very elegant mechanical steps. First, we cut. We use those restriction enzymes to digest or cleave both our donor DNA and our delivery vector. Second, we align. Those enzymes purposefully leave behind complementary sticky ends. Thanks to the natural chemical attraction of DNA base pairs, these overhangs want to snap together like perfectly matched puzzle pieces. It's honestly a bit of molecular magic. And third, we seal. We add DNA ligase to permanently glue those sticky ends together, locking in our new recombinant DNA molecule. But why go through all this trouble? Well, once we put that molecule into a host cell, our manufacturing objectives basically fall into 3 real-world buckets. 1, massive gene cloning, where we build huge research libraries to study specific diseases. 2, protein recovery. This is literally how we turn host cells into biological factories to pump out synthetic insulin for diabetics today. Or 3, functional integration. This is where we embed a gene to express a physical trait, like engineering crops that naturally fight off pests. If our goal is that third objective, a new physical trait, we head into Section 4, transforming cells and plants. We need a reliable pipeline to deploy this code into a complex living system. This plant transformation pipeline is a rigorous 5-step journey. We start with introduction and integration into the plant cells, then move through expression and regeneration of the whole plant. But I want to really emphasize step 5 here, transmission. This is the holy grail, right?
Transmission means the transferred gene is successfully passed down to the plant's offspring. The genetic edit becomes permanent and self-replicating for all future generations. That is the true lasting power of this technology. Having established the foundational hardware, let's jump to Section 5, the CRISPR Precision Upgrade. This represents a breathtaking leap forward in what we can actually do.
This is the ultimate software upgrade for genetic engineering. Traditional recombinant DNA relied on cutting and pasting foreign genes, right? But modern advancements treat the organism's existing DNA as directly programmable code. Systems like CRISPR-Cas9 bypass those old sticky ends completely. Instead, they use guide RNAs that act like a highly advanced GPS, navigating billions of base pairs to find one exact location. It's literally like upgrading from a pair of blunt scissors to a word processor, where you just hit find and replace. This pinpoint precision is exactly how we're starting to cure previously untouchable genetic conditions, like sickle cell disease, by correcting the native code directly. When we compare traditional rDNA to modern gene editing side by side, the shift is staggering. We've moved from relying on bulky biological vectors, like plasmids, to directly delivering ribonucleoprotein complexes, or RNPs, using CRISPR. For human therapeutics, this is an absolute game changer. Why? Because RNPs do their editing job and then just degrade naturally. They don't leave permanent, potentially dangerous viral vectors hanging around in the human body. We've gone from forcing totally foreign traits into organisms to safely repairing specific native mutations. Because of these advancements, scientists today select their toolkit based on the specific challenge. If we need massive bulk production of a foreign protein, like churning out insulin, we still use the traditional rDNA factory. It's a fantastic, cheap assembly line. But if the challenge needs subtle, targeted tweaks within an existing organism, we switch to the CRISPR pipeline. CRISPR acts like a bespoke, microscopic surgeon. It is perfect for those exact, localized adjustments where you need absolute accuracy. All of this brings us to our final section, the arc of innovation. Let's zoom out and look at the larger trajectory of these breakthroughs. Our timeline traces a pretty wild journey. We started with simply discovering the DNA double helix. Then we figured out recombinant vectors, those cut-and-paste traditional tools we talked about. And now we're squarely in the era of programmable edits with CRISPR. Just think about that for a second. The leap from merely discovering what DNA looks like to actively programming it with microscopic precision happened in less than a single human lifetime. The speed of this innovation is just mind-blowing. And that perfectly sums up the core theme here. From the initial discovery of the DNA double helix to the programmable precision of modern gene editing, the history of genetic engineering is the relentless pursuit of perfect biological control. We've spent decades sharpening our tools, moving from crude splicing to elegant programming, all to master the biological blueprint of life itself. So I'll leave you with this to ponder. We now hold the power to cure debilitating diseases and secure our global food supply. With CRISPR giving us the precision to literally rewrite the blueprint of life, the big question isn't how we edit the code anymore. The technology is already here. The real question is, as we achieve perfect biological control, what exact future are we going to build with it? Thanks so much for joining me for this explainer. I hope it gave you a fresh perspective on the wild world of genetic engineering. Keep asking those big questions, and I'll catch you in the next one.